It survived 5 mass extinctions and humanity might kill it. The horseshoe crab has lived unchanged for 450 million years. Dinosaurs came and went. Trilobites vanished. 96% of all marine species died in the Permian extinction. But this creature remained exactly the same. Not similar. Not evolved. Identical. On a beach in Delaware Bay, thousands of horseshoe crabs crawl from the ocean. They've made this same journey for millions of years. Same moon phase. Same tides. Same beach. A female drags herself onto sand. 4 males follow. She digs a nest and deposits 4,000 green eggs. The males fertilize them. Then they all return to the sea. This scene played out before humans existed. Before mammals walked on land. Before the Atlantic Ocean even formed. The horseshoe crab is older than trees. Older than sharks. Older than Saturn's rings. But here's what nobody tells you. This ancient survivor has blue blood worth more than gold. Biomedical companies harvest 600,000 horseshoe crabs every year. They drain 30% of their blood. Then they return most of them to the ocean. 30% of those crabs die within days. The blood saves human lives. Every vaccine, every injectable drug, every medical implant gets tested with horseshoe crab blood. Without it, modern medicine collapses. But the harvest is accelerating. And for the first time in 450 million years, horseshoe crab populations are crashing. What makes their blood so valuable? And why can't we survive without it? The horseshoe crab's design is perfect. Evolution stopped trying to improve it 420 million years ago. While other species adapted and changed and died, this creature found a formula that worked. And never deviated. 4 species exist today. Limulus polyphemus lives along the Atlantic coast of North America. 3 species inhabit Asian waters. They all look nearly identical to fossils from the Paleozoic era. The same dome-shaped shell. The same 10 legs. The same long tail spine. They're not actually crabs. They're chelicerates, more closely related to spiders and scorpions. They have 10 eyes scattered across their shell. 2 large compound eyes. 5 additional eyes on top. 3 eyes underneath. Each eye type sees different wavelengths of light. Scientists at the Marine Biological Laboratory in Woods Hole, Massachusetts studied their vision for decades. They discovered horseshoe crabs can detect ultraviolet light humans cannot see. But their eyes revealed something shocking in 1956. A scientist named Keffer Hartline won the Nobel Prize studying horseshoe crab vision. He discovered how neurons process visual information. The breakthrough came from this ancient creature. Human eye doctors still use principles learned from horseshoe crab eyes. Their blood is even stranger. It's blue because it uses copper-based hemocyanin instead of iron-based hemoglobin. When oxygen binds to it, it turns bright blue. But that's not the valuable part. In 1956, scientist Frederick Bang at Johns Hopkins University made an accidental discovery. He injected bacteria into a horseshoe crab. Within hours, its blood turned into a solid gel. The blood had detected the bacterial toxin and neutralized it instantly. Bang realized the blood contained special cells called amoebocytes. These cells detect endotoxins, the poisonous substances produced by gram-negative bacteria. This wasn't just interesting. It was revolutionary. At the time, pharmaceutical companies had no reliable way to test for bacterial contamination. They used rabbits. Inject the rabbit with a drug sample. Wait 48 hours. Check if the rabbit developed a fever. The rabbit test was slow, expensive, and unreliable. Bang and his colleague Jack Levin developed the Limulus amoebocyte lysate test. LAL test. They extracted the blue blood. Isolated the amoebocytes. Created a reagent that would gel in the presence of endotoxins. The test was 100 times more sensitive than the rabbit test. It gave results in 45 minutes instead of 2 days. By 1977, the FDA approved LAL testing for all injectable drugs. By 1987, it became mandatory. Every vaccine you've ever received got tested with horseshoe crab blood. Every antibiotic injection. Every 4-fluid bag. Every medical device that touches your bloodstream. The biomedical industry needed horseshoe crabs. Needed their blood. And needed a lot of it. But nobody asked if the crabs could survive the harvest. The harvest happens in facilities along the Atlantic coast. Commercial fishermen catch horseshoe crabs during spawning season. May and June. They load them onto trucks. Drive them to biomedical laboratories. Inside Charles River Laboratories in South Carolina, workers strap horseshoe crabs onto stainless steel racks. Rows of crabs hang upside down. A technician inserts a needle into the membrane around the heart. The bright blue blood flows into collection bottles. Each crab loses about 30% of its blood volume. That's roughly 100 to 150 milliliters. The crabs hang for 24 to 72 hours. When the bottles are full, workers remove the needles. Load the crabs back onto trucks. Return them to the ocean within 72 hours of capture. The industry calls this process humane. They claim mortality rates are only 15%. But peer-reviewed studies tell a different story. A 2020 study by the University of New Hampshire found actual mortality rates between 18% and 30%. Some crabs die during transport. Some die during bleeding. Most die after release. Female horseshoe crabs that survive the harvest show even more disturbing effects. They produce fewer eggs the following season. They're weaker during spawning. They often cannot make the journey up the beach. Biologist Gary Creamer tracked bled females for 3 years. Their reproductive success dropped by 57% compared to crabs that were never harvested. The industry harvests 600,000 crabs annually in the United States alone. At a 25% mortality rate, that's 150,000 dead horseshoe crabs every year. Just for their blood. But here's the midpoint shock. The blood harvest isn't even the biggest threat. Commercial fishermen catch 1.4 million horseshoe crabs every year for bait. Eel and conch fishermen cut them up and use them in traps. They're not harvested for medical use. Not preserved for research. Just ground up for bait. Because eel and conch prefer horseshoe crab meat over anything else. Between the blood harvest and the bait fishery, humans kill or severely harm 2 million horseshoe crabs every year. For the first time in 450 million years, their population cannot keep up. Delaware Bay once had the largest horseshoe crab population in the world. In the 1990s, surveys counted 1.2 million females. By 2019, that number dropped to 335,000. A 72% decline in 20 years. The crash triggered an ecological disaster nobody predicted. Red knots, a species of shorebird, migrate 9,000 miles from Argentina to the Arctic every spring. They timed their arrival at Delaware Bay to coincide with horseshoe crab spawning. The birds gorge on horseshoe crab eggs. In 2 weeks, they double their body weight. Then they continue north to breed. But when horseshoe crab populations crashed, red knot populations crashed. In 1982, 95,000 red knots stopped at Delaware Bay. In 2020, only 23,000 arrived. The red knot is now listed as threatened under the Endangered Species Act. Because the horseshoe crabs are disappearing. Scientists call this a cascade effect. Remove one ancient species and the entire ecosystem collapses. 14 species of shorebirds depend on horseshoe crab eggs. If the crabs vanish, the birds vanish. If the birds vanish, the insects they control explode in population. The cascade spreads. And the biomedical industry keeps harvesting. In 2003, scientists in Singapore developed a synthetic alternative. They called it recombinant factor C, RFC. It was genetically engineered to detect bacterial endotoxins exactly like horseshoe crab blood. Same sensitivity. Same accuracy. No crabs required. Dr. Bao Ho at the National University of Singapore led the research. His team isolated the gene responsible for endotoxin detection in horseshoe crab blood. They inserted that gene into insect cells. The cells produced factor C protein. When mixed with endotoxins, it produced the same clotting reaction as LAL. The test worked. In blind studies, RFC matched LAL results 99.7% of the time. The European Pharmacopoeia approved it in 2016. Japan approved it in 2020. The technology existed to replace horseshoe crab blood entirely. But American pharmaceutical companies refused to switch. The FDA approved RFC in 2018 but only as an alternative to LAL, not a replacement. Companies could use RFC if they wanted. They didn't have to. Why? Money. The LAL industry generates $0 million annually. 5 companies control the entire market. Lonza, Charles River Laboratories, Associates of Cape Cod, Waco, and BioEndo. They own the horseshoe crab bleeding facilities. They own the LAL patents. They own the distribution networks. Switching to RFC would require new equipment. New validation studies. New regulatory submissions. The upfront cost scared companies away. Even though RFC is cheaper long-term. Even though it's more consistent. Even though it saves horseshoe crabs. A 2020 investigation by The Atlantic found that pharmaceutical companies resist RFC because they fear regulatory delays. The FDA approval process for switching from LAL to RFC takes 2 to 3 years. For a company launching a new drug, that delay costs hundreds of millions in lost revenue. So they keep using horseshoe crab blood. Keep harvesting 600,000 crabs per year. Keep driving populations toward collapse. Because changing is expensive. In 2022, the pharmaceutical company Eli Lilly became the first major company to switch entirely to RFC for its injectable drugs. They announced 0 horseshoe crabs would be harmed in their manufacturing process. Activist groups celebrated. But Eli Lilly is one company. Hundreds of others still use LAL. Meanwhile, Asian horseshoe crab populations face even worse pressure. China and Japan harvest 3 species for both biomedical use and food. Horseshoe crab soup is considered a delicacy. The eggs are eaten as a traditional medicine. The Chinese horseshoe crab, Tachypleus tridentatus, is now critically endangered. Surveys in Hong Kong found only 200 breeding adults remaining in 2021. That's down from 50,000 in 1990. A 99.6% decline in 30 years. The mangrove horseshoe crab, Carcinoscorpius rotundicauda, faces the same fate. Vietnam and Thailand have declared it commercially extinct in their waters. It still exists but in numbers too small to sustain fishing. The Japanese horseshoe crab, Tachypleus gigas, is listed as endangered. Its habitat has been destroyed by coastal development. Only a few breeding populations survive in protected bays. 3 of the 4 horseshoe crab species on Earth are now endangered or critically endangered. The creature that survived 5 mass extinctions is being driven to extinction by the species that depends on it most. The horseshoe crab survived 450 million years because it adapted perfectly to its environment. Humans have existed for 300,000 years. In that geological blink, we've pushed this ancient survivor to the brink. This isn't just about horseshoe crabs. It's about what they represent. The Permian extinction killed 96% of all marine species. The Cretaceous extinction killed the dinosaurs. The horseshoe crab walked away from both. But it cannot walk away from industrial harvest, coastal development, and climate change happening simultaneously. Scientists call our current era the 6th extinction. Human activity is driving species extinct at 1,000 times the natural rate. We're losing biodiversity faster than any time since the asteroid that killed the dinosaurs. And we're doing it to creatures that survived everything else Earth could throw at them. The horseshoe crab teaches us a dark lesson. Survival isn't about being the strongest or the smartest. It's about not being valuable to humans. The moment we discovered their blue blood could save human lives, we started killing them. The moment we discovered their meat worked as bait, we started grinding them up by the millions. Conservation groups are fighting back. The Ecological Research and Development Group in Delaware runs breeding programs. They collect horseshoe crab eggs from beaches and raise them in protected hatcheries. After a year, they release juvenile crabs into the wild. Survival rates increase from 0.03% to 10%. But they can only release 150,000 juveniles per year. We kill 2 million adults. The Atlantic States Marine Fisheries Commission sets harvest quotas. In 2019, they reduced the allowable catch in Delaware Bay by 50%. But enforcement is weak. Commercial fishermen routinely exceed quotas. And the biomedical harvest has no quota at all. Companies can take as many crabs as they want. The solution exists. We have synthetic alternatives. We have breeding programs. We have the science to save them. We lack the will. Because changing costs money. The horseshoe crab survived the Ordovician extinction 444 million years ago when glaciers covered the continents. It survived the Devonian extinction 375 million years ago when ocean oxygen collapsed. It survived the Permian extinction 252 million years ago when volcanic eruptions poisoned the atmosphere. It survived the Triassic extinction 201 million years ago when supercontinent Pangaea split apart. It survived the Cretaceous extinction 66 million years ago when an asteroid ended the age of dinosaurs. But it might not survive us. That's what makes this the darkest story of all. The creature that proved survival was possible became valuable to the species that makes survival impossible. If you want more stories about Earth's forgotten survivors and the industries destroying them, you'll want to see what we uncover next. One question remains: what other ancient survivor are we harvesting into extinction right now without even knowing it? Send this to someone who needs to hear it. Then subscribe for a new dark truth every day.